In this section, we will look at genetics. We will talk about genetics and probabilities, test crosses, complete dominance, incomplete dominance, and codominance. It is important for us to realize that probability and chance play central roles in genetics. Probability has a central role in genetics for two reasons. The first has to do with the law of segregation. The second reason is that fertilization is a chance event. If we think about the law of segregation, we know that any gamete produced by an individual that is heterozygous has 50% chance or probability of carrying a dominant allele and a 50% chance of carrying the recessive allele. This means that the offspring has a 50% chance of receiving the dominant allele and a 50% chance of receiving the recessive allele. We can use these probabilities in conjunction with the other parent's probabilities and gametes to determine the probability of getting an offspring with certain genotypes. Here's an example. If we look at a male who is heterozygous for albinism, so big A, little a, and a female who is homozygous and an albino, little a, we can ask, what is the probability that their child will also be an albino? We first must look at the gametes from each parent. The father is heterozygous, so has a big A, little a. This means that in his gametes there is a 50% chance of having a gamete or a sperm cell with a big A and a 50% chance of having a sperm cell with a little a. In the mother, her genotype is homozygous recessive, so because these alleles are segregated, there's a 50% chance of having the first little a in an egg cell and a 50% chance of having the second a in an egg cell. Because these little a's are indistinguishable, we can say that there is 100% chance that the female will pass on a little a to her offspring. To figure out the chance of having a child that is homozygous recessive, we have to multiply the probabilities from each parent. So there's a 50% chance, or 1/2 chance, of having a little a from dad, and a 100% chance of having a little a from mom. So this means there is 1/2 or 50% chance of these parents having a child with little a, little a. Here's another picture of that scenario. The mother giraffe is albino, so there's a 100% chance of her producing eggs with little a's. The father is heterozygous. There's only 50% chance of him producing a gamete with little a. We multiply those chances or probabilities together, and we see that there's a 50% chance that offspring will be albino. Punnett Squares will give us these same probabilities in a slightly different format. We can start with our cross, the heterozygote times homozygous recessive. We can set up our Punnett square. We can put parental genotypes on either side, and this Punnett square helps us with segregation as well. So we are separating out the big A and the little a. We're going to put dad's genotype on the side of our Punnett square. We will put mom's genotype, little a, on the top. From here, we fill in each of our boxes, and this will give us possibilities that we can see in the offspring. So, for each box, we're going to take the letters, top and bottom, from mom and dad, and put them into the box. So, each box represents a 25% chance of that particular genotype showing up in the offspring. In this case, we see that there are two boxes that have little a, which means half the boxes have little a, so there's a 50% chance of having an offspring that is homozygous recessive. Here's another example we can work through. Tay-sachs is a recessive disorder if both parents are carriers or heterozygous for Tay-sachs. What is the possibility or probability that they will have a child with Tay-sachs? Tay-sachs is a recessive disorder. This means that if the parent has a big T, they have a normal phenotype. If they have two little t's, we will see Tay-sachs. So we can make a little key of traits or alleles to the side to help us keep this straight. Each parent is heterozygous, so they have one big T and one little t. This means that when they produce gametes, there's going to be a 50% chance of their gamete having the big T and a 50% chance of their gametes having the little t. Both parents are heterozygous, which means each one has a 50% chance of a big T, 50% chance of passing on the little t. If we multiply together these probabilities, 1/2 times 1/2, we see that there is a 25% chance that the child will be homozygous recessive and have Tay-sachs. We can also do this cross in a Punnett square. So our cross is one heterozygote crossed with another heterozygote. We are going to put the parental genotypes on each side of our square. So dad on the side, mom on the top. We then fill in the Punnett square, and we will see the possibilities for the offspring. In this cross, only one box has little t, little t. This means that there is a 25% chance of having a child with Tay-sachs. In Chapter 7, Part 1 lesson, we learned that just because you know the phenotype of an organism does not necessarily mean that you know the genotype. Sometimes the dominant phenotype can have two possible genotypes. They can be homozygous dominant, or they can be heterozygous. Here's a scenario with this exact problem going on. We're working in a zoo, and you would like to produce white alligators in a mating program. You have a pigmented alligator, and the problem is you don't know for certain the genotype of this alligator. This alligator may have a homozygous dominant big M genotype, or they might be heterozygous big M, little m. In either case, the phenotype is for normal coloration. So, how can we figure this out and determine what the actual genotype for our alligator is? To do this, we can perform a test cross using an individual with a homozygous recessive genotype, which means we cross our alligator with the unknown genotype with an albino alligator. To write this, we know that our alligator has one dominant allele for pigmentation. We are unsure of the other allele, so we will leave that blank. We're going to cross that alligator with an individual that is little m, okay, coding for albinism. To run this test cross and determine what is going on, we will use a Punnett square. So our cross so far is big M blank times little m. We can use a Punnett square and fill in what we know to get started. Dad's genotype is big M blank. Mom's genotype is little m. We can fill in the top half of our Punnett square, big M, little m, big M, little m. And we know that mom will always give little m's to the offspring. From here, we have to look at the offspring to determine what is actually going on. There are only two possibilities. If the father is homozygous dominant with two big M's, we will see that all of the offspring will have a heterozygous genotype. So 100% of the offspring will be pigmented. If the father is heterozygous with a little m, we will see that 50% of the offspring will be pigmented and 50% will be albinos. Okay? This mix tells us what the genotype actually is. So if we look back at our alligator, we have run a test cross. We have our pigmented male alligator, and he has been mated with a female albino. The clutch of babies hatches out, and we see both pigmented and albino babies. So, what is the genotype of the father? Because the albino babies show up, we know that he has to be a heterozygous. For albinos to show up in the offspring, the dad and the mom both must have little m's to give to the offspring. So far, all of the problems that we have been working on have been with complete dominance. This means that the dominant allele completely masks or hides the recessive allele. Let's work through another example. Here is our next problem. Brown fur is dominant to white fur in rabbits. An individual with a homozygous dominant genotype is crossed with a recessive white individual. What is the predicted genotypic ratio of the offspring? What is the predicted phenotypic ratio of the offspring? So this first problem is a case of complete dominance. Our first step is to make ourselves a key of alleles. So brown is dominant, so big B will represent our brown allele. White is recessive, so our lowercase b will be the white allele. Okay, so next thing we need to determine is our actual cross. So blank times blank. What are our parents? So an individual with homozygous dominant genotype. So that means that homozygous is two of the same alleles, and we're talking about the dominant allele. So the first parent is big B, big B. Okay, the recessive allele is the little b, so a white individual is going to have two copies of the recessive allele, and that's the only way to get that white coloration to show up. Okay, so once we have our cross, we're going to divide or segregate our gametes and put them on a Punnett square. So we'll draw our Punnett square. Okay, so first parent is big B, so we're going to put a big B here and a big B there. Second parent is little b, so we'll put those gametes on the top of our square. Once we have that, we're going to fill in our square. Okay. So one gamete from each parent. And then we have to go back to the problem and answer what is actually asked. So this problem wants to know the genotypic ratio, which is what letters are actually in our box. So the genotypic ratio is 100% big B, little b, or heterozygous. So the phenotypic ratio is going to be a translation of our genotypic ratio. Okay, so what color are these rabbits? So big B, heterozygote has one dominant allele, which means that all of these rabbits are going to be brown. Here's another practice problem. What is the possibility that a cross between a pigmented giraffe, who is homozygous dominant, and an albino giraffe, homozygous recessive, will yield albino offspring? This second problem is very similar to the first. So we're talking about pigmentation versus albino. Okay, so a pigmented giraffe, let's use big A for pigmentation. And we'll use little a for albino. So albino is the recessive allele or recessive trait. And so our parents, we set up our cross, we have a homozygous dominant giraffe, big A, crossed with an albino giraffe. And the only way to see the albino allele show up is to have two copies of the recessive, so little a. So put those gametes on our Punnett square. First parent is big A, second parent is little a. We fill in the box with one allele from each parent. Okay, then we go back and figure out what this problem is asking. So we're asking, will they yield an albino offspring? So if we look at our box, all of these are big A, little a, heterozygous, and as long as there is a copy of the dominant allele, these giraffes will have pigmentation. So there's a 0% chance of having an albino baby. Not every gene shows complete dominance. We are going to look at two other cases. Incomplete dominance is when the heterozygote appears to be an intermediate between the two homozygotes. So, in these flowers, we see that there are homozygous red flowers and homozygous white flowers. If you mix those two and have a heterozygote, we see that the offspring will be pink. Another case is codominance. This is when the heterozygote displays characteristics of both homozygotes. This means that both alleles are equally dominant and both will show up in the heterozygote. So in this case, with these cows, we have a homozygous white cow, a homozygous brown cow, and in the heterozygote, we see both brown and white spots. Let's compare the three types of dominance. We're going to use the example of flower color in plants. The capital R will be the red allele, the lowercase r will be the white allele. For complete dominance, if the genotype is homozygous dominant, the individual will be red. If there's a heterozygote, they will also have red flowers, and homozygous recessive will have white flowers. So in this case, the dominant allele completely hides that recessive allele. If we look at incomplete dominance, a homozygous dominant individual will still have a red phenotype. The heterozygote will be a mix of the two, so they will be a pink flower, and a homozygous recessive will still be white. For codominance, homozygous dominant will again still be red. Homozygous recessive, little r, will still be white. And our heterozygote will show both red and white petals. Let's practice with incomplete dominance. In this case, we have black coats in dogs are incompletely dominant to yellow coats in the Labrador Retrievers. The intermediate heterozygous condition results in a brown or chocolate lab. So we see this is incomplete dominance because the dominant allele, the black coat, is not completely masking the yellow coat or lack of pigmentation that we see in the homozygous recessive. This results in sort of a blending of the two colors and gives us the chocolate or brown colored lab. This question asks, a brown lab is crossed with a chocolate lab. What is the genotypic and phenotypic ratio of the puppies? To start this problem, we need to make a key of alleles. In this case, we're going to use the letter B. So big B will be black and little b will be yellow. A brown lab is crossed with a chocolate lab. This is two ways of saying the same phenotype. Brown, chocolate, same thing. So we said before that brown means that we have a heterozygous individual. Okay, so big B, little b, crossed with a chocolate lab, which is also brown, so another heterozygote. And so now we've got our parents. We're going to put their gametes on a Punnett square. So there's our Punnett square. We've got big B, little b for parent number 1, big B, little b for parent number 2. Now we can fill in our square. So big B, big B, big B, little b, big B, little b, little b, little b. Okay, so the question asks for both the genotypic ratio and the phenotypic ratio. So genotypic ratio is reading the genes. So what are the actual ratios of the letters in the Punnett square? So in this case, we have 1/4 of our babies are big B, 1/2 of them are heterozygotes, big B, little b, and 1/4 of them are homozygous recessive. For phenotypic ratio, we're going to be looking at the actual colors of the dog, so what we see, the physical features. So in this case, we have to translate these big B's, little b's into an actual color. So big B is going to be black. 1/4 of our boxes have the big B, so we have 1/4 black puppies. Half of the boxes have big B, little b, so those are brown dogs. And the last 1/4 of the boxes are little b, which means that we're going to have yellow labs. With codominance, we see that 2 alleles are both dominant. Neither one is masked by the other. So in this case, we're talking about dogs that are black and white, and both of these colors are dominant. The first thing we want to do is set up a key of our genotypes. So with both incomplete dominance and codominance, that is a crucial part of our problems. For this problem, we're looking at black dogs and white dogs. So big B is going to be a black dog. Our heterozygote, big B, little b, is going to show up with both types of spots, black and white. Lastly, our little b is going to be white. So for this problem, we're looking at a black and white dog, so the heterozygote, crossed with a solid black dog. It's going to be big B. Once we have our cross, we're going to put our gametes into a Punnett square. So big B, little b for parent number 1, big B for parent number 2. We're going to fill those in with the appropriate gametes. Okay, this question is then asking what is the chance of getting a puppy who is solid black. So now we need to look inside of the Punnett squares and see which genotypes correspond to black individuals. So if we look back at our key, we know that big B is the black puppy. Okay, and there are 2 boxes with big B. So to answer the question, we have a 50% chance of having puppies who are black. In this lesson, we have covered probabilities and their importance, and how we can use Punnett squares to figure out these probabilities and phenotypic and genotypic ratios. We've looked at complete dominance, incomplete dominance, and codominance in these organisms.